Hi, good morning, everybody. Great to see you all back today. So without further ado, I'd like to open this session on infrastructure, support public health. We have 4 great speakers and some great panelists to discuss all of these great initiatives, implementation initiatives, and think about sustainability of and equity of providing this infrastructure to the public health community. So without further ado, I'd like to invite Peter van Heusden to the stage, who's going to be speaking about Not All Those Who Wander Are Lost, PHA4GE Infrastructure Working Group, Guidance for Growing Public Health Genomic Sequencing Efforts. Thanks, Peter.
Hello, everybody. The title, to some extent, is a reference to the fact that we've been working for several years without actually publishing the guidance that we have come up with. But let's start off with what people ask us and what we don't really want them to ask us. So do not ask us which server to buy. Okay, this is, as somebody who's worked in computational infrastructure for years, this is the number one question. You know, I've got a grant, I need to buy a server, which one should it be? And instead, when we started discussing this thing, because as the infrastructure working group, myself, Danny Park is my co-chair, others in the group, we come from very different contexts. We are working with data at different scales, we are supporting different communities, and so then we kicked off just a few months, as with the rest of PHA4GE, before the start of the COVID-19 pandemic. And then when the pandemic started, then many people got involved in genomic sequencing that hadn't been before. And then we realized that the crucial question should be like, so how is your analysis run? How are you running your data analysis? How does the data flow into your analysis, genomic analysis process, and from there to where it gets used? And who has access? What does the access lifecycle look like? And how much of this does your own infrastructure need to solve? So, as I said, we came from very different perspectives. One thing, however, became clear. How does the analysis run is a question whose answer is converging. So if we look at like what some options are, you know, then in the past we've seen these kind of mega scripts, bash scripts that people put together to do the data analysis. In fact, I remember Torsten sharing a 10-line, Torsten Seemann, because there's 2 Torstens in the room, sharing a 10-line bash script for how he analyzed SARS-CoV-2 data, which was the basis of the methodology that was basically used thereon. But then, I mean, we know that they generally are not portable, often have hard-coded paths, the dependencies are not well specified. So then what we've seen is the emergence of workflow languages. Workflow languages and containers are the 2 technologies that have been transformative. So different container engines, you know, Aptainer, Docker, Singularity, we're fairly agnostic, but what they do is they basically pin your dependencies in place in a way that becomes independent of the underlying operating system. Then around that, the cloud of workflow languages, Nextflow, Widow, Galaxy, CWL, SnakeMake, that abstract away the problem from the specifics of what you run, the specifics of how you run things to the general statements about what you want to run. And really supporting that, there are both workflow creation communities, so IWC in the Galaxy world, NF Core in the Nextflow world, and then workflow repositories, so Docstore and workflowhub.eu. And so what we said, what we kind of converged on is that how you should run your workflow is using containers, using a workflow language, preferably using a workflow that has been published and reviewed by a group of reviewers on a resource like this. So at least we've got an answer for one thing. The rest depends. My colleague Kim Ng from Start Serum Institute in Denmark put together a beautiful set of case studies that we still use as basis for discussion within the infrastructure working group that looked at the journey of a lab from succeeding in its first sequencing up until truly massive scale. And what you do depends on where you are on this spectrum. So I want to look at two case studies. The one Placide already mentioned yesterday, which is the evolution of sequencing of Ebola in the DRC. And then the other one from, supported by PHA4GE, but the California Department of Public Health's COVIDNet system. So if we look at what Placide presented yesterday, then, I mean, as he said, they have unfortunately had several Ebola outbreaks. They have extensive experience in responding to them, and I mean, I mean, the way that they've grown their capacity over the years has been truly inspiring. But now with the most recent Kasai outbreak, what did they do? They deployed very rapidly a pre-configured laptop with the software that they needed. We saw the software mentioned in Placide's slides. It happens to be a containerized workflow language pipeline. This got deployed to the outbreak zone, and they were able to do very rapid sequencing. The laptops are many African labs have powerful laptops and sometimes desktops because when they're doing nanopore sequencing, then they'll have a machine with a GPU and some beefy processing power just to do the base calling. So then a machine like that can get repurposed for bioinformatics. It works in a low internet context with experienced bioinformaticians that know which commands to run on the command line. Although I think they said they were using Epitome Desktop, but anyway, that's generally how it works. And it's ultra portable, okay? So this is, look at the scenario here. This is a small number of priority samples that you need to process as fast as possible. The other end of our matrix of possibilities, COVIDNet, which was near real-time SARS-CoV-2 sequence analysis at a massive scale. So we're still trying to figure out exactly, because of privacy reasons, we can't exactly pin down how many of these sequences were from California, which happens to be, by the way, the state of California has a GDP which would make it the 4th richest country in the world. So this is not a low resource setting. But it extensively makes use of cloud-hosted platforms, which basically means that they take away the need to worry about the managing of the computational layer themselves, the infrastructure layer. They barely need to use DevOps because they can just knit together different Google resources. So it uses the Terra platform, it uses workflows created by PHA4GE, and it uses Looker for the dashboards. And I mean, this is processed more than 420,000 samples. I mean, a truly massive scale. And I mean, that's the graph on the bottom left there is just the viral pipeline of PHA4GE being run. You can see the peak of sequencing activity during the pandemic. But because this is a generic platform, it can run a workflow, okay, any workflow you give it. This has been repurposed. So we now see that the second row there is bacterial workflow. So we see the same groups that came up to speed with being able to process viral sequences now shifting on the same infrastructure to bacterial sequence analysis. So the key point here is to find the common themes in your workflow construction that you can exploit to be able to pivot between different things. And by the way, Joel Savinsky is in the room, so he can answer any of your PHA4GE and Terra questions if you've got them. And I mean, as you know, Placide is around as well. So when we look at these infrastructure choices, besides scale, they're always a trade-off. We're working in public health. This is often a regulated environment. Risk is a problem. You can have infrastructure that minimizes risks and is fully compliant with all of the data requirements of your broader IT system. You can have cutting-edge research software because bioinformatics is a very fast-moving field. And you can have small maintenance footprints. And it's really tough doing all three at once. Although I must admit, from ISO in a box, we're trying to make it easier to at least do this, but that's another talk, not this one. And when you are facing these trade-offs, you have to decide, what do you want to manage yourself? So are you going to manage all of your infrastructure? Maybe you need to because of data sovereignty concerns. I mean, most groups that I've seen are managing some degree of local infrastructure because some things don't fit well in the cloud. For instance, exploratory analysis, you can run up a cloud bill very, very quickly. Long-term data storage as well. Are you using a platform as a service like Terra or Galaxy, where the platform handles most of the basic infrastructure deployment, but then you have flexibility in terms of workflows? Or are you using software as a service, where basically you know that what you're doing is a pre-solved problem, and you're just going to provide the same simplified interface to some analysis pipelines that you're going to run over and over again? And I mean, or are you using some combination of these things? So take-home messages from our working group are that there are a few recipes. When we started, we wanted to actually come up with, here's an Ansible script to configure your servers or something like that. We discovered that because infrastructure is embedded within people's context to such a large degree, there are very few direct recipes. I suspect that actually this is also why from the Pipelines and Viz working group, we're seeing lots of talk about pipelines, a little about Viz. Visualizations is similar to infrastructure in this way in that it embeds itself so tightly with other data systems and other data consumers that we don't see as much directly reusable visualization dashboards as we see reusable pipelines. I mean, and remember that when we're saying reusable pipelines, that's doing the bioinformatic analysis. The broader data analysis problem, as we said, the data flow, where it comes from and where it goes to, that is a larger question that we often are not seeing completely, okay? Solutions come from the intersections of many competencies. When you're starting off, the people that you need are going to perhaps have some set of skills. As you grow, you're going to need people with new skills. Assuming that one person can do all of those things is a good way to lose staff because they're going to be tired of you just adding to their workload and they will leave. And reflection and policy matters. Infrastructure is a policy question. Be deliberate about how you manage a data lifecycle, etc. So, and then finally, we as a field don't talk to as infrastructure builders, don't talk to each other nearly enough, which is why this is a great opportunity, but also join the infrastructure working group so that you can be part of future conversations. And finally, we do get asked, which server do you buy? And here's a slide answering that. I'm not going to speak to this slide, but what I will say is was developed for Africa CDC to look at what configuration makes sense in terms of minimizing the IT load in the IT maintenance load in different contexts. And thank you to so many of my members of the working group, Danny, Kim, Karin, Nabil, Tom, Joel, David, Soyen and Dwani and many others who we've learned from.
Thanks, Peter. It's amazing work I think the Infrastructure Working Group does, and commend all of the effort they put into thinking about these problems. So we'll have Q&A for the 4 speakers at the end. So as yesterday, you can store your questions up. But next, I'd like to invite Chaoran Chen to the stage, where he's going to be talking about Gen Spectrum dashboards for genomic epidemiology.
All right, good morning. So in my talk, I would like to talk a bit about a very database-driven approach to analyzing data and building dashboards. And to illustrate the underlying idea, I'd like to start with us imagining or thinking about how amazing Lego robots are. If you can come up with an idea for a design for a robot, you can just build it yourself. You don't need a big lab, you don't need a big team. You can just realize your own idea for a robot very easily. And this is possible because very much thanks to this piece in the middle, the engine. And while you can come up with thousands of different ideas for robots, from pipetting robots to cars, you just need to use the same engine over and over because it is very reusable and flexible in what it can support. And this is an approach we are also following in Gen Spectrum. by developing a database engine. Interesting background. This was not intended to be the background. The background was supposed to be white, by the way, so some text might be a bit different to read. I don't know what happened. So yeah, the idea here is we have a database engine which is flexible in the queries it can support, and we use it for various dashboards, including our own curve spectrum and gene spectrum dashboard, but also for example, Localus and Patoplexus, and also other third-party tools. So what is a database? And we have been hearing about databases already a few times at this conference, but when I talk about database, I mean it in this case in a more fundamental way. So to quote Edgar Cott in his 1970 paper, who is basically the inventor of modern-day databases, he wrote that future users of large data banks must be protected from having to know how the data is organized in the machine. And it provides a basis for high-level data language, which will need maximum independence between programs on the one hand, and the machine representation and organization on the other. And translating this idea to our case, it means data analysts and data scientists should be able to do their work efficiently without needing to worry about data structures, data storage, data formats, or the performance. And we try to make this possible for genomic epidemiological data. So we developed a software called Lapis and Silo. It is a database software for viral genomic epidemiology. And in other words, it is a software that facilitates the use of genomic epidemiological data without needing to know and worry about how the data are organized in the machine. And the core requirements here is that the database must be scalable to at least tens of millions of viral sequences, and it should be fast. So Lapis and Silo support 3 different types of data. The one type is just metadata, and that's like any existing conventional database already support. It's a table of data with different types of data like numbers, text, or dates. But then what makes our database special is the support for additional data like the sequence alignment and phylogenies. In case of sequence alignment, in principle, it's also a table. It's a very, very large table with tens of thousands of columns, where each column basically has one character. But conventional databases are not good at all at handling this. Databases can maybe support 1,000 columns, but they cannot support 30,000 columns. So what we did is to make it possible to support this type of data very efficiently. And also, since recently, we support the storage and querying of phylogenies, where you can have, for each of the sequence, you can link them together in a tree topology, and also provide the corresponding tree branches and branch lengths. So here are just a few examples what you can do, what you can ask the database. So you can use it to find the most common recent ancestor of sequences of a particular lineage, let's say A2, with an arginine at position 120 in the S protein. Or you can ask in which countries have sequences with at least 5 of the following mutations be found, and then provide a list of mutations. Or you can ask which mutations have been found in sequences from Europe in the past months, and what are the proportions. And in all those cases, you can formulate those questions in a high level or easy way. You don't need to worry about the data structures, and about looping through lists, hash maps, etc. You just ask a question, and the database, it's up to a database to optimize the response. And we use this database engine for our dashboards. So we have our gene spectrum dashboard, which supports a wide range of pathogens, including SARS-CoV-2, influenza A and B, RSV, Mpox, dengue, CCHF, measles. And we also support that for each of the pathogens, list of different views. So you can look at the sequencing efforts view if you just to see who in the world is doing a sequencing, how much is being sequenced, where are the sequences coming from. But you can also analyze individual variants or compare different variants in different views. On the dashboard, you can filter the data, of course. You can filter them by different metadata, country, dates, sampling strategies, host, etc. And you can also filter by the variant. You can filter by the lineage or mutations, combinations of mutations. And in the background, the work is not done by the dashboard itself. It's all done by the database engine. And we then have a list of different views. For example, we have the phylogenies since recently. Hereby we store a very big phylogeny, ideally with all the samples that are there. And we use for visualization Taxonomium, developed by Theo Sanderson. And for COVID, we downloaded the Usher tree from the Usher team with millions of tips. And what you can do on the dashboard is you can filter by different countries or filter by mutations, and then subsample basically or filter down the tree for only samples with those mutations. And then you can also look at the tree in detail or color them by different metadata fields. It is possible then to see, of course, a number of different other plots. So, for example, you can see the number of sequences, or you can see a map with the sources of the sequences. And here, all the different components are reusable. So they are like the Lego bridges and stones. You can use them to build your own dashboards if you like. So to conclude, a database should not only provide access to the data, to the raw data, but I think it's very important that we build databases that make it really easy to use the data. Especially if the amount of data expands, it will get harder and harder for just an individual person to analyze big amount of data, and it would be great if you can decouple the use of the data by just formulating what you want to get from how you want to process and to get to the data. There's still a lot of data not incorporated in the system. For example, raw data, raw reads. I mean, there are a ton of raw reads available, but they're, of course, very, very big. And often they're just lying in either some archives or disk storage of the labs. And it'd be great if you can make it possible to easily query and access those data as well. And of course, there's much more: protein models, results from immunological studies, cases data, and so on. And then lastly, we are of course often asked, can this be expanded to bacteria data? And we are very interested in this. We are trying to look into those data at the moment, but of course it has quite some challenges with much more diverse data and different genomes. So with that, I would like to close and acknowledge and thank the whole team, the whole CEVO group, in particular also my supervisor, Tanja Stachner, and our funders from the US CDC, ETH Zürich, and Swiss universities. Thanks.
Thank you, Chaoran. Really exciting work. And so moving on to our next speaker, Karin Lagesen, where she'll be talking about transdisciplinary collaboration and data democratization for genomic pathogen surveillance. Thanks, Karin. Please come.
Good morning, everyone. I'll want to start to mention that my title sounds high level, but all of this is actually about infrastructure, because the infrastructure is the thing that makes these things possible. So first, a little bit about my institute, the place I work. The Norwegian Veterinary Institute has a One Health focus, and we work closely with our counterpart, the Norwegian Institute for Public Health. For pathogens, our institute, we handle samples from food and animals, while the NIP handles those from humans. A core part of our mission is to provide independent research-based advice to the government. We're a national reference lab for several pathogens, and as such, it's our responsibility to keep track of these pathogens and know about their occurrence and their current characteristics. We're a fairly small institute. We're around 350 people, and we have people of very diverse backgrounds. Not surprisingly, we have quite a few vets, but we also have microbiologists, epidemiologists, statisticians, and more. For genomics, this means that any system we set up has to be able to accommodate people with very different computational experience. So at the NVI, we started working with genomic data in the early 2010s. We were initially research focused, and we built batch-style pipelines on the national compute infrastructure. As genomics became part of our routine surveillance, our needs changed. And as many other people, we started building Nextflow pipelines, but we soon realized that this approach didn't really fully meet our needs. The clue was that the people who were most interested in the surveillance results from these pipelines were rarely the ones who could or wanted to run the analysis. Now, having bioinformaticians run the analysis for them did help a little bit, but we were still limited in our ability to interpret the results in context. And that's the clue when it comes to surveillance, to have that work properly. So that raised a question. How do we bridge the gap between computational workflows and biological insight? A common approach is to make biologists more computational, or vice versa. But in practice, stretching people too far outside of their fields of interest, that frequently doesn't really work all that well. So instead, we realized that what we really needed to do was to rethink the entire model. What we wanted was a system where both sides could play to their strengths. And traditionally speaking, what many people do is that they have sort of a core facility set up to do these things. In this setting, the bioinformaticians process the data and hand the results over to the domain experts. What we wanted to do was to reverse that. We wanted to enable the domain experts to explore and understand the data on their own terms, while still having the bioinformaticians organizing things in the system. So to achieve this, we realized we needed some software that could help us bridge this gap between the two worlds. After evaluating several options, we settled on Irida, a system that's developed by some Canadians who are, well, some of them are in this room, but yeah. Irida allows biologists to explore their own results, while bioinformaticians focus on maintaining the pipelines and ensuring good information flow in the system. To complement the Irida system, we also chose to set up Clarity LIMS, so that's the Illumina Laboratory Management Information System, to manage the lab workflow. Aside from making life easier for the people in the lab, it has also significantly contributed to the information flow of our system. When people submit samples for sequencing, they add information that is then carried through the entire system and help decide which analyses are done in the end. So I just want to add that setting up this system, even when we've used existing software, has required quite a lot of local adaptation. We've built our own LIMS workflows and our own bioinformatics platform, but not only to manage the daily analysis and automation, but also to satisfy requirements on what analysis we need to do, what outputs we need to produce, and on tracking samples and information, and also the reporting we need to do to various instances. So having these systems up and running has been quite beneficial for us. All of our data is centralized and in an interface that enables people to explore their data and make decisions and to do their own analysis. Main feature of this is the line list data view that you can see here. This is very familiar for anybody who's ever used Excel. And this familiarity has really helped us in adoption among people with diverse computational skills. And this means that our users are less reliant on others to access their data and to do analysis. They're capable of going into the system and having a look for themselves. So, for a couple of lessons learned, one of our biggest realizations have been that the best tools for research might not be what's the most useful for surveillance. An example here is our AMR pipeline. We use RESTfinder for this because EFSA, the European Food Safety CDC, has standardized on a specific version of the tool and the database. We are mandated by law to report AMR every year, and they have specified this tool and this database, and thus that's what we need to have. We do want to get AMR Finder Plus up to have more up-to-date results, but we haven't gotten there yet. Another thing we've learned is that we have to be very careful and deliberate about updates of our databases. Surveillance is a key activity for us, and that means that we do need to keep things consistent over time. Back to the AMR question again, that means that whenever we do update our underlying AMR databases, we do need to rerun all of our old isolates, just to make sure that an absence of a gene isn't simply a symptom of that gene being absent in the database when we run that particular isolate. And this brings me to the final part of my story. How did we manage to get here? In short, by doing a whole lot of talking. We've spent a great deal of time figuring out what the different people do, and more importantly, why they do the things they do. This has not always been an easy process, but it's been essential for us to understand what's going on and how we can make the system work in practice. And when having such conversations, one thing that needs to be remembered is the difference in background. I think many of us have been in a situation where you've heard two people talk and you've barely understood anything. They might as well have been speaking in code. And that's because they are from the same discipline, they have the same background, they're very high up in shared knowledge. The further apart two people are in disciplines, the further down in the knowledge stack you need to go to find a shared ground. More of the basic things have to be explained. And that can be a major challenge when trying to learn from each other. And that brings me to my last and perhaps most important learning point, teaching point. Learning from each other requires a certain mindset. It means that you need to dare to be ignorant. You need to be able to step out of your own comfort zone and try to look at things from somebody else's perspective. Sometimes it's not even enough to have somebody explain things. Sometimes you have to look at their screen with them and see what they're clicking on and figure out why they're clicking on it. It's also very important to dare to ask questions, even about things that seem obvious. And equally important, the person being asked needs to remember that the question, why did you do that, is actually not meant as a challenge. And that can be a very hard instinct to overcome. And through this process, it's important to remember the main point of it all, and that's to create a shared mental model of what's going on so that all of the pieces can work well together. And with that, I'd like to acknowledge some of the people I've been fortunate to have these kinds of conversations with. So as you can see, this is a very diverse set of people that I've collaborated with, and this is not all. And I'd like to acknowledge my institute and also Elixir Norway for helping me to come here today. Thank you.
Thanks, Karin. I think lots of great messages reminding us that accessibility is an important role in turning data into impact. So finally, I'd like to welcome Clare Sloggett to the stage. And she's going to be talking about building AusTracker, lessons learned from 6 years of deploying a national genomic data sharing and surveillance platform.
Hi, everyone. So I'm going to be talking about AusTracker, and just for context, the Australian context where we built AusTracker is that we have state-based health jurisdictions. So we have 8 different states and territories. They own the data in general. And when there is an outbreak investigation or we want to have a national surveillance effort, obviously pathogens don't respect state boundaries. And this has traditionally been one of the issues people have had to wrestle with. It's almost like coordinating surveillance between countries. So in response, there was an effort. The conversations actually started back in 2015. It took several years to get these governance committees and processes set up. It was a major effort. I won't really have time to talk about that at all. But there is an AusTracker poster, which I think is number 35. And I encourage you to go and talk to Tiet, who will be with that poster if you're interested in that. So that effort was ready just in time for us to have a pandemic. So the very first version of AusTracker was built in a great hurry to handle the COVID data, and then there's been kind of continual development since then. So what we have now is a surveillance platform where the model is that the different jurisdictions upload their sequence data and their contextual sample metadata. The analysis is carried out by a trusted national analysis team, and the governance and the analysis team are both distributed. They have members across the jurisdictions. And then the analysis results and the appropriate contextual metadata is made available back to the jurisdictions, but the sequence data is not, right? So they don't have to share that directly with one another. So this is our basic model for the way the data flows through the system. The public health labs over on the left there can upload sequence data and contextual sample and sequence metadata through either the web interface or through a command line interface. They get real-time validation as they upload the data, which helps us to keep it clean. So just basic things like the dates in the right format, and if you have a controlled variable, is it one of the allowed values? So that really helps making the data usable, that it is validated at the point where it is uploaded. The data they put into the system is something they still own. I should probably have said this, part of the reason we built this is that some of these variables are things that people are currently not comfortable putting into global public repositories. So we needed a semi-private system, at least at this stage. So they own all this data. We don't own any of the data. They can choose to share it with one or more projects, or 0 or more projects, really. And a project, these green boxes on the right, could be an ongoing surveillance effort, or it could be a particular outbreak investigation. And they are configurable so that only the approved metadata variables are going to appear in any particular project context. So you won't necessarily see all the available sample metadata in a particular project view. And not every jurisdiction may be participating in every investigation. The access to those is role-based, so we have a kind of a quite granular individual role-based access system to all the different powers and all the different pieces of data. The sequence data is not made available to the participating jurisdictions, but it is put on is synced across to the analysis server, where the analysis team will analyze it, and the results will go back into the project context. I'm just thinking if there's anything else I needed to say that. All right, sorry. So at the moment, this is hosted on the Azure cloud, and the analysis server is on-prem, just because we found it more cost-effective to do the analysis on-prem. But we are working towards a fully containerized version where the whole platform can run anywhere, because we would like this to be useful to other people. We'd like it to be able to run potentially in countries where there is no Azure cloud node or no AWS node or whatever else. So just to give you, I won't have time to go through the whole system, but just to give you a quick feel for what the system looks like. People can submit data through the web interface. It looks like this. They can just drag and drop a CSV file or an Excel spreadsheet, and they will get back validation messages in the browser. They can also submit sequence data in the same way. So on this screen, I've drag and dropped 6 FASTQ paired end Illumina files in, and it's kind of detected that it looks like 3 pairs and given me a chance to correct it if it was wrong. And we also have this command line interface, which again, I won't go through at all. I just want to say that it exists. To people who are used to command line tools, it's got a kind of familiar hierarchical command structure. It really covers all aspects of the system. We use it for administration. The thing that it's most often used for by most users is for submitting data. So some people would prefer to put metadata or sequence data in via the command line. And it hits the same API, so the responses you get back will be exactly the same in either case. And we found this was well worthwhile writing. It actually makes development easier to have a command line interface. The effort is absolutely worthwhile. So slightly ambitiously in a short talk, I'm just going to play a quick video of what some of the visualization aspects look like in a project view, just to give you a feel for it. Not everything. So when you log into Austracker, you only see the projects in which you have an appropriate role. You don't see all the projects in the system. So this would just be a handful of them. I'm not sure that's playing. There we go, thank you. All right, so this particular account would just have access to a handful of projects. This one I'm showing is just a demo project. It's not real data. Each project has a dashboard, which is like a real-time snapshot of what the data in the project currently looks like, and these are configurable per project. Oh, sorry, I think we've gone forward. We have. Yeah, so each project gets a dashboard. These are just a real-time view of what's currently available in the project. Could you play this video? Maybe guys, thanks. There's also a sample line list, so just a tabular view of the available metadata, which you can search and sort and filter and export and do all the things. So this will contain the rows are the samples that have been shared by the contributing organizations. The columns are going to be the approved metadata variables only, as well as some of the columns will be derived from the analysis carried out by the analysis team. So for instance, the CGMLST values. We also have interactive plots, which again have exactly the same metadata available. We've just added maps. We're building on those at the moment to add more features to those. And we have a bunch of just standard plots that you might expect. So for instance, this one is an epidemiological curve. It's got all the same variables available which you could configure to be viewed on those plots. So here I'm coloring it by CGMLST, which would have come from the analysis, and then faceting it by the jurisdiction that contributed it, just as an example. And finally, the last thing I'll show you is we have this interactive tree viewer. So the results of the phylogenetic analysis, again with the same metadata variables available for view on the tree. These trees have various features, which I won't have time to go through all of them, but we could put the metadata variables into the leaf labels, or we could use them to color the nodes, or we could put them in as kind of metadata blocks, like a heat map style view against the tree. And every tree has an attached sample table. Anything you select on the tree will be selected in the table and vice versa. So this is a nice way to do a metadata search for the exact cohort of samples you're interested in and then find them on the tree. And we also have some nice navigation features. So just there, I just jumped to the smallest subtree that contains the samples of interest. And we had to do this really early on because the COVID trees were big, right? People were not going to navigate those manually. We needed navigation features. Okay, so getting towards the end of time. So I just want to put up some of the lessons learned through this development process. When we started out, we thought we were going to be concentrating on tree visualizations and bioinformatics pipelines. And it turned out to be lots of other priorities that we kind of hadn't anticipated and lots of other parts of the effort. So in our particular context, the data access and the permission system was a big deal because it just really helped to build trust. If you had this role-based access and people knew exactly who would be allowed to see what, it made them much more willing to share data. So we spent a fair bit of time on that system. There was probably, although we're on the cloud, there's a lot of cloud expertise required for the kind of system that we were deploying. We were lucky in that we had a person, I've said dedicated DevOps, but actually half software developer, half DevOps engineer, so we were very lucky there. We spent more time than we probably expected on just user support and data management, chasing things up. There's not the data in this project that we expected. Did you share it with the one you intended to share it with? Did you share it at all? This kind of thing. I think I said before but the kind of modular architecture that we had has been very useful because the system has no opinion on what the pathogen is or what analysis is going to be run, right? It's just a system. So everything is configurable and the pipelines can pull the data and then they can push the results and we can design the dashboards and we can configure the variables, right? Even the metadata fields are not baked in, right? We configure them on the fly. Scalability, we started during COVID, so we thought about scalability from the start, but there is this continual choice you have to make between implementing a feature that will work right now and maintaining scalability for the next pandemic, right? Because that's what we're meant to be ready for. And just finally, you've seen a lot of talks about data standards and ontologies. Because we started in a pandemic, there was not a PHA4GE standard for SARS-CoV-2 at the point where we started. It was just a list of fields, and it's grown from there as we've added more pathogens. So one of our goals is to organize those fields and do a bit of a reorganization of those. But we have tried to maintain compatibility with external standards wherever possible, at least on the conceptual level. So there's like a one-to-one mapping between the fields, and we kind of know what those are. All right, and I'll be very happy to talk to anyone who is around. Thank you for your time. I think I'm right at time.
Thank you, Claire. Can I invite all of the speakers back to this stage? Peter, Chaoran, Karin. And we will open it up for a brief Q&A, a couple of questions. So please wait for a mic. Please introduce yourself. But yeah, we're open for Q&A, so please. One down the front here, please.
Hello, this is Gültekin. I would like to thank all the presenters for their presentations. And my question is to Karin. So she mentioned about the Veterinary Institute and having a lot of analysis on the background, having a diverse people on the background, working a lot. So I wonder, how do you use that data to inform the public health policies? Like, how do you connect with the people from the human health side, and how do you inform these policies and like AMR and other things? How do you collaborate? Because I think it's really hard to collaborate with different entities. So I would like to hear your experiences.
Well, one of the things to keep in mind here is that sequencing data is in many ways a very new thing. And that is something we're seeing a lot in Norway. So Norway, the way things are organized, it's very sector-based. We have the public side, the human side, and then we have the veterinary side. And we're even under different ministerial departments. So that's how high up the divide goes. And one of the things is that any sort of, there is no legislation that really covers sequencing data. And this is especially challenging for the human side because the only rules and regulations they have basically say that they should take any data related to humans and lock it down in a drawer, basically. So they're currently working a lot to be able to allow to be able to share something, but the way the system works now is that we share with them in very concrete outbreak-related cases. We don't have any formal sharing agreements yet, simply because this is so new. There is no regulation, there is no legislation. We're doing it because it works. But a lot of this is currently working on a trust basis. We're a tiny country. We're 5 million people. And half of us, most of the people in this field, know each other quite well. So we know who to call. If you have a salmonella outbreak, you know who to call on the other side. But this is, it works, but it's also slightly uncomfortable because we don't have formal systems to fall back on. And that is a challenge, and we're hoping very much that legislation both in the EU and nationally is going to help open up these things a little bit more so that we can get more of a routine way of sharing and collaboration going on this, so that we can help make sure that everybody is safe and happy, basically. Thank you.
Thanks, Karin. Any follow-up questions? Another one down the front.
Hi, this is David Yuan from ENA. I have a question for Clare. I was just curious that whether, by the way, thanks for the good overview introduction of the OS tracker. I was just wondering that if there are any regulations or considerations at this point to generate revenue from the tracking system, so that you can feed back to the communities that are providing the samples, implementing care and share principle to a certain extent.
Yeah. So the data, I'm not sure if you mean something more specific by feeding back to the community, but the data that the jurisdictions put in is data they own, and they see the results of that analysis. But because they are the owners of the data, it's really up to them, I guess, what can go out beyond that to the community. In the other direction, one of our goals, one of the things we don't do yet, but would really like to, is to help people to chaperone their data into INSDC, where they've decided that that's appropriate. But did you mean something more specific by going back to the community?
I was mean something more specific. Let's say that a research project goes to Aboriginal community, collecting the samples, and that kind of information can be really valuable or eventually generate some revenue for, let's say, make the medicine.
Yeah. In this case, it's all public health management. So there is actually a research program hosted, which is the Oz Pathogen program, that is research into public health. So I think it's really up to the data owners. It's not up to us to decide what can be done. Yeah.
Okay, thank you.
All right, thanks. And for the sake of time, we're going to move on to a different panel discussion now. But I'd like to thank all of our speakers again one more time.